So, as I was saying, bacterial cell has its own structure and are forming cellular organelles that enable these cells to function well. This is a diagram showing you the bacterial cell and the various structure that makes the cell to work. We have the flagellum. Many of you know the function of the flagellum. There are many organisms that possess flagellum. When they have two or more, we call it flagella. It is actually used for locomotion. Send some messages here. Hold on. You don't need to type your name for attendance. You don't need to type your name for attendance. Attendance is taken automatically. So now this is a diagram. This is just like a pictorial representative of the bacterial cell. If you look at the bacterial cell from the microscope, it's not going to look like, but this is a representative of what the bacterial cell looks like. So we will be looking at these structures one by one. The most important one for this class, I will look at their functions subsequently. The bacterial cell structure is divided into 4 parts. We have the surface layer, which includes the cell wall, cell membrane. And we have the appendages. We have the cytoplasm and the special structure. Now, the surface layer of the bacterial cell consists of those are the surface layer, those are the surface parts. This includes the cell wall, this includes the cell membrane, and also the capsule. Now, these are, please stop writing your name at the chat. You don't need to type your name. Don't type your name. Okay, no problem. Type your name, please. Type your name, type your name, type it, type it, type it. Type your name, okay? Type your name, it's all right, but there's a way the attendance is captured, okay? You want to type your name, but there's a way the attendance is captured. If you type for somebody, it will definitely not bring out your, so it's very, be careful. If you are typing your name, it's going to show is your name. If you type for somebody, this is electronics, you will be flagged because it will show. So if you want to type your name, type your name. It's all right, okay, it's all right. But ensure that you don't type for somebody, because if you're typing for somebody, it's just like a chat. When you write maybe like Anna and Belu, and that is your name, and you are writing another name for somebody, it means Inkem. So you put yourself in trouble. Type your name, ignore those who are not present. Let's move on. Now, the surface layer consists of those cellular structure at the external part of the bacterial cell, which we have the cell wall, the cell membrane, and the capsule. Now, the appendages are those structure that protrudes out of the cell, such as flagella, pili, or fibrae. They are the appendage. They form kind of, we can call it the leg in quotes Let me use that word. Bacteria doesn't have legs, but that is kind of their legs, their hands. For animals, for insects, their appendages are actually their legs and their other hands and all that. So we have cytoplasm. Cytoplasm is the internal part of the bacterial cell that consists of the materials that are enclosed, that are not seen on the outside. The surface layer can be seen in the microscope, the appendages can be seen in the microscope, but the cytoplasm is covered, is the internal structure, which is the ribosomes, the mesosomes, nuclear material, which is DNA, the RNA, inclusions, body, the plasmids, and what have you. These are all embedded in cytoplasm. And also, we have bacteria that possess special structure. The special structure for bacteria is actually the endospore. So we'll be looking at these structures, like I said, one by one, give definitions to them, look at their functions, and how do they help the bacteria carrying out its normal activities. Cell wall. The cell wall is the outermost part of the bacteria, which consists of some interlinked molecules, and it gives the cell a definite shape and structure. It provides structural support for the cell. It also protects against infection and the mechanical stress. It separates interior of the cell from the outer part of the membrane, outer environment. It also enables transportation of substances and information from the cell internal part to the exterior part. And it also allows substances to be transported from the exterior to the interior. That is the semi-permeable membrane that serves as barrier. We call it cell wall. So we know the function of the wall. The wall prevents or protects. It protects the internal organ and it prevents the external from coming in. It also helps in osmotic regulation. It helps in osmotic regulation. That is, it allows the cell to regulate its temperature. And it helps to prevent water loss. It helps to prevent water loss because it has a way of conserving the internal fluid. Excuse me. So it helps to prevent water loss because there are times the bacteria find itself in an environment that is very harsh and the bacteria may lose water. So the cell wall helps to prevent water loss. It helps to conserve the water and also perform osmoregulation. It also prevents the cell from rupturing due to tug-up pressure. Tug-up pressure is as a result of influx of fluid into or outside the cell. So inside the cell, when there is too much fluid inside, the cell will rupture. So it prevents act as barrier. It aids in division of gases in and out of the cell. So there is an exchange of gases and it passes through the cell wall. The cell wall serves as a medium for gaseous exchange. And this allows the bacteria to carry out the respiratory function. It also provides mechanical protection from insects and pathogens. Yes, bacteria can be a pathogen, but bacteria is also a prey to some other pathogens. Unfortunately, we are all muted. I would have asked you what is the predator for bacteria. I can ask you to unmute yourself and tell us if anybody knows what is the predator that normally hunts bacteria. Can anybody tell us? I said it provides
Bacteriophage.
bacteriophage. That's good. Bacteriophage is a virus that can kill the bacteria. So bacteria also avoid bacteriophage from killing it. Now, it provides this mechanical protection from other insects. There are other insects that also try to invade the bacteria. So the cell wall serves as this. Now let's look at the picture of the cell wall. Now, this is a picture of the bacterial cell wall. We have the gram-positive cell wall and the gram-negative cell wall. I believe you must have been taught these differences between gram-positive cell wall and gram-negative cell wall. So the gram-positive cell wall has a larger portion of the cell with peptidoglycan, and the gram-negative has a slim layer of peptidoglycan. This is one of the major differences between the two cells, okay? So if you look at the bacterial cell wall of gram-negative bacteria and gram-positive bacteria, you can see some differences. For gram-positive bacteria, it is not complex, it is very simple. It has the cell membrane and also the peptidoglycan. While the gram-negative cell wall has what we call the outer membrane layer. It has the outer membrane layer, then it also has the slim peptidoglycan, and it has a wider periplastic space and other lipoproteins. It is more complex than gram-positive cell wall, though they have similarities such as the peptidoglycan and the periplastic space, though one is bigger than one and we have similarities. Now, the cell wall also allows bacteria to carry out cell-to-cell communication. The physiological and biochemical activity of the cell wall helps in what is called quorum sensing. It is a cell-to-cell communication between two bacteria. You can see that we have, let me show you this, okay? Hold on. Can you see this one here? This here, they are bacteria. Now they are communicating. This is the communication link between them. Can you see my marker? So this is where they are communicating at the cell wall. So it allows them to carry out what is called quorum sensing. Quorum sensing is a cell-to-cell communication between 2 or 3 or more bacteria. Quorum sensing is like you making a phone call. As a human, you make phone call to your cells. Microorganism has a way of communicating. Why do they communicate? If they are in a danger zone or they are in a zone that is probably needing reinforcement, they do a cell-to-cell communication that is called quorum sensing. This quorum sensing allows them to begin to sense the need for microorganisms to accumulate in a particular region. This accumulation allows them to form what is called a biofilm. It allows them to form a biofilm. And when they form a biofilm is a community of microorganisms. It could be diverse community, it could be single community. Diverse means we have different types of microorganisms. We can have E. coli, we can have Pseudomonas aeruginosa, we can have Staphylococcus. They all form a biofilm. The biofilm looks slimy. It's like a thick community of bacteria. If you have seen the way some spirogyra grow around a drainage, that's a biofilm. A biofilm is slimy. The slimy formation of a bacterial community. Now, this cell-to-cell communication allows them to generate signals that attract bacteria from one end to another. It normally happens during infection. Whenever a bacteria is at a spot where it's causing infection, then the cell will allow the bacteria to make communication to other bacteria of its kind. So they generally locate the infectious bacteria of its kind and begin to accumulate. Because before a bacteria can actually invade a cell, there is need for inoculum site. Inoculum site is the total number of the bacteria that is formed at that particular concentration. So the inoculum size must be sufficient enough to form an effective dose that is causing damage at a particular site. So a single bacterial cell is not sufficient to form infection. It needs accumulation of other bacteria. And this is one way by which microorganisms sense the need to form a colony or biofilm at a particular site. So the cell wall helps the microorganism to do what is called cell-to-cell communication. Don't forget that. Let me remove it out. So I think I still have some explanation on the slide on this. Bacterial cell in biofilm communicate in a density-dependent manner called quorum sensing. I've explained that before. This is an animation that shows you what I'm talking about. Cell-to-cell bacteria use quorum sensing to coordinate certain behavior, such as biofilm formation, virulence, and antibiotic resistance based on the local density of the bacterial population. So this animation you see here, one bacteria is telling the other bacteria that, come see what I got. Look what I just ate. Say, girl, I'll tell the rest of the guys to keep a lookout. There could be more. So the bacteria that is invading the macrophages and the T cells, informing the other bacteria that they can come and have a feast. That's exactly what crop sense is all about. It's just like when human beings make a phone call. So, hey guys, let's go and have a party here. Everybody comes, driving. That's exactly what's happening here. So bacterial crop sensing is a way of communicating between bacteria, and they form a biofilm. Biofilm is very resistant to antibiotics. It's very resistant to sunlight. It's very resistant to certain factors, environmental factors, because it's a community of bacteria, millions of cells. Now, this is the difference between gram-positive and gram-negative bacterial cell I was explaining the other time. Both of them possess peptidoglycan, but the gram-positive cells has a larger part of the peptidoglycan, while in gram-negative cell, there's a slimy layer. Now, for the gram-negative cell, we have additional structure, which is the outer membrane, which is not present in the gram-positive cell. And also, this space is for virulence, periplasmic space, which is also not conspicuous in gram-positive. So let's just say it is not present. In some cases, you can find it, but in most cases, it is not conspicuous, but it's highly conspicuous in gram-negative cell. So this forms a major difference between gram-positive and gram-negative cell. If somebody asks you, what are the major differences between gram-positive and gram-negative cell? It is not only peptidoglycan. We have the outer membrane, we have the periplasmic, and the peptidoglycan. They form a major difference between the two cell walls. This is the chemical structure of the cell wall. Chemically, it is the part of the cell, which is the peptidoglycan. It is made up of two hexyl sugars and acetylglucosamine, which is NAG, and acetylmuramic acid. It is a cross-linked, interconnected peptide chain. It's like a wire, but it's cross-linked. If you look at it, we have the NAM, which is the acetylmuramic acid, and the NAD, which is acetylglucamic acid, glucosamine, which they are interwoven together in alternate form. They can be in tri, tetra, or pentapeptide chains. So they form the meshwork, MESH, which form the part of the cell. In gram-positive cell wall, like I said, they have the thicker peptidoglycan. They also possess polysaccharides, which is tetroic acid, which is polymers of glycerol and ribitol phosphate. Some gram-positive bacteria, such as Mycobacterium, contain lipid and mycolic acid, while in gram-negative bacteria, they possess lipopolysaccharides and periplasmic space, and also the outer membrane. So let's look at another structure, which is the mesosomes. Mesosomes is another structure found in the bacterial cell. It helps in cell division. The way the bacteria divide is by binary fission. So they help in bacterial cell division. They also aid cell wall synthesis. If you remember, the cell wall, it consists of NAG and NAM. So all these chemical substances have to be synthesized. They have to be synthesized from ribosomes to form a strong cell wall. So this allows a cell wall to be strong. The mesosome also possesses another function, which is the aid in respiration. They aid in respiration of the cell, and they also help in secretion of processes. Secretion of processes means there are some processes that are being carried out in the cell that need some enzymes. So they help in carrying out the function. They also increase the surface area of the plasma membrane and also the enzymatic content. These are the functions of the mesosome. It helps in cell division, they aid cell wall synthesis and DNA replication. They also aid in respiration, secretion processes. They also increase the surface area of the plasma membrane and the enzymatic content. So, another structure of interest is the ribosome. Ribosome is a very important structure in the bacterial cell. It's a very important structure. It's critically important because many microorganisms, let me say, virtually all microorganisms need ribosome to function. Ribosomes are cell organelles that consist of RNA and proteins. This is the structure responsible for protein production. You recall that every developmental processes in the cell requires protein synthesis. And protein synthesis is carried out in the ribosomes. They are responsible for assembling the protein in the cell, and they are responsible for converting, for housing the conversion of the DNA codes, converting it to the language the cell can understand, which is the protein, and therefore expressing it by carrying out what is called protein synthesis and also cellular repair. Ribosomes are typically composed of two units, two subunits, a large subunit and a small subunit. We have ribosomes in eukaryotic cell and we have in prokaryotic cell. In eukaryotic cell, ribosomes are 80S, while prokaryotic ribosomes are 70S. They are synthesized in the nucleus and they cross over the nuclear membrane, the cytoplasm, through the nuclear pore. Let's look at another function, which is a cell capsule. The cell capsule is a very large structure in prokaryotic cells, most especially bacteria. Our main focus is on bacteria. Some of what we are talking about applies to fungi. Some of them applies to protozoa, but our main focus is on bacteria. So we are looking at the context of bacteria. It is a polysaccharide layer that lies outside the cell envelope of bacteria and is thought being part of the outer envelope of a bacterial cell. It is found in the outermost part of the cell. It is like the external part, capsule. It's from the word capsule. If you ever taken an antibiotic capsule, you know the content is actually like a powder and it's shielded by two cases and close together. When you open it together, when you open it up, you see the powdery antibiotics that you want to take. And this antibiotic that you are swallowing is actually the powder. If you put the powder, remove it and put it on a spoon and swallow it, it will still function. The capsule is just a delivery medium in drugs. It has no drug effect. It's going to dissolve the moment it gets into the water. Now, bacterial capsule functions such because it protects the bacterial cells from external forces. If you open up the capsule of the bacteria, you see the bacterial cell inside. So the bacterial capsule, though, does not have any contribution to its growth, but it has some of the function for the bacterial cell. It is a well-organized layer, not easily washed off. and it can be the cause of various diseases. So bacterial capsule can actually be a virulent factor. When we say virulent factor, that's a factor that makes an organism to be able to cause disease. So bacterial capsules are actually an added advantage for every bacteria. It allows the bacteria to be protected and allows the bacteria to cause disease. Because most capsules are so tightly packed, they are difficult to stain using standard stain because most stains cannot adhere to the capsule. If you remember your gram stain, when you carry a gram stain, if a bacteria does have a capsule, it remains invisible during your gram stain and during your microscopic view because the capsule is difficult to stain. It does not allow stain to stick to it. There's a special staining for capsule, okay? So when capsule of a bacteria is observed under the microscope, it forms a kind of layer where we look at it. This is what I'm talking about. This is a bacterial capsule and a slime layer. It shows under the microscope the capsule of the bacteria. Look at this picture very well. This is a gram stain, and you can see this transparent form around the cell. It shows that the bacteria possess a capsule, though you can't see the capsule very well, but it gives that indication. It's a capsule because it does not allow a penetration of the stain, especially into the cell. So what are the functions of the capsule? The capsule is considered a virulent factor because it enhances the ability of bacteria to cause disease by preventing phagocytosis. When a bacteria gets into the cell, the first thing is there is an interrogation by the cell defensive mechanism, such as the macrophages, the T cells, and other defensive mechanisms. We have the white blood cells, and we also have the interferons that normally prevent disease infection. Now, when the bacteria cell enters, the bacteria is seen as an antigen. These antibodies fight against it by engulfing it. The mode of feeding of these macrophages is by engulfing. The way amoeba normally feed, they engulf the bacteria. Now, because of the capsule, it prevents it from being engulfed through phagocytosis. And when it is not killed, it goes on to carry out its dangerous activities, which leads to disease. For those who don't recall, phagocytosis, phagocytosis is the process that the body uses to get rid of unwanted things within the body. So things like harmful bacteria and foreign bodies are removed from the cell. The capsule can also protect from engulfment by eukaryotic cells, such as macrophages. I've said that. Capsule also contain water, which protects the bacteria against desiccation. During desiccation, when there's no water, the capsule contains certain level of water and it helps to conserve it for the bacterial cell. So it helps the bacterial cell to utilize the remaining water during drought or desiccation. They also exclude bacterial virus and most hydrophobic toxic bacteria, such as detergents, out of bacterial cell. Capsule also helps cells to adhere to surfaces. It helps the bacterial cell to stick to surfaces. And when they stick to surfaces and the surfaces are clean, the bacteria remains clung to that surface, and thereby it can cause disease, especially if those surfaces are medical equipment that are used for surgery. Medical equipment that are used for surgery must be thoroughly cleaned to remove bacteria. When bacterial capsules are glued to medical device, just ordinary washing cannot take care of it. You have to do what's called sterilization. And I want to believe you have been taught what is called sterilization. It is only a sterilization procedure that can actually exclude bacteria from such surfaces. Inclusion bodies. Inclusion bodies are also organelles that are found in the bacterial cell. They are surrounded by a thin non-union membrane that separates them from the cytoplasm. Their main function is for storage. There are a wide variety of inclusion bodies in different types of cells. In prokaryotic cells, they are mainly formed to store reserved material. In eukaryotic, they are used to store sugar, lipids, and others. So they are nuclear or cytoplasmic aggregate or stainable substances, usually protein. They typically represent sites for viral multiplication in a bacterium. Now, when a bacteriophage infects a bacteria, the site of viral multiplication is usually the inclusion body, because the inclusion body is like a storage facility that helps to reserve materials needed by the cell. It usually consists of viral capsid protein. The inclusion bodies have a non-unit lipid membrane. So inclusion body is a very important organ, such as we have important capsule, we have cell wall, we have ribosomes, we have the mesosomes, and those we have discussed today.